Hey everyone, in this video lecture we're going to be learning about cladograms. So a cladogram is a diagram that we're going to use to visually represent the evolutionary relationships between different species or different taxonomic groups of organisms. You can think of it sort of like the tree of life, showing the branches of how different species evolved through time. And there's two ways that we can create these cladograms. It's either going to be based on shared traits amongst the organisms, and this is sort of historically how cladograms were created, looking at what traits organisms have in common. The more traits they have in common means they're probably more related to one another. But more modern day, since we have the technology, cladograms nowadays would be based on genetic information. So we'd look at DNA and compare how many nucleotides are the same or different, and we can use that to construct these trees. But for this video, cladograms we're going to really think about being based only on shared traits, but note that if we want to make it more accurate, we could use genetic information. So trying to understand the components of how to construct a cladogram, lines on cladograms represent time. So in this case, the way it's laid out, the horizontal lines would be evolutionary time, the leftmost point would be the sort of oldest time, and the rightmost points on the lines would be more present day. Where you see branching, which are indicated by the little yellow dots, branches indicate there's a common ancestor that eventually split into two distinct groups. So that would be showing where that evolution over time is occurring. And at the very tips of the branches are where you'd put the present day species that you're trying to compare to determine who's more related to who. And there are different formats that you can draw a cladogram in. So there are 3 different cladograms on the screen here. All 3 are identical in the way that they're comparing organisms. It's just different formatting. So the one I showed you is where the time is horizontal, so old to present goes from left to right, and the present day species are on those branch tips on the right hand side. You can basically flip this and have the oldest time point at the bottom and the present day at the top. Again, same exact way we're comparing these species, we've just flipped it. And then sort of different formatting is, instead of using more right angled brackets when you're drawing your branches, you just do straight lines that make it look more V or Y shaped. Again, all of these are the exact same cladograms, just different formatting. So if we want to understand how to interpret a cladogram, the distance of the species on the cladogram is important, because the closer their species are to one another, the more closely related they are evolutionarily. The more physical distance between them on the cladogram, the less related they are. So if we were to compare primates and rodents to rodents and fish and need to determine which pairing is more closely related, we would literally track the distance on the cladogram. So if we go from primate over to rodent, pretty short distance. If we go from the rodent and follow its pathway all the way to fish, there's a much larger distance between those two groups. So in terms of who's more closely related, it would be the primates and fish. There's less evolutionary time between them, indicating they're more closely related. We also can look at cladograms to figure out who has what traits. So where a trait appears in evolutionary time, any organisms after it are gonna have the trait. Organisms before it will not have the trait. So if we look at the trait of amniotic egg, it sits right here on the main line. So in evolutionary time, this is where the trait appeared. So any organism after this point is going to have the trait. Any organism before would not. So organisms that do have it would be the turtles and birds, because this point happens after the amniotic egg. We can also go up here. Rodents would have it, as well as primates. Again, they are all after that point in time where the amniotic egg trait appears. So now you kind of understand how to read them. Let's think about how to construct them. I would do a little bit of practice in class where we practice interpreting these cladograms, but let's first understand how we can construct them. So we're basing this on shared traits only, not DNA. So this first step is to take the species that you want to compare, and you need to identify traits that they have in common. And so if these are the organisms I want to compare, I want to compare a frog to a rodent, to a lizard, to a gorilla, to fish, to bird, I need to find traits that go from kind of most common to least common. So I want to compare vertebrae to having 4 limbs, to having the amniotic egg, to eggshells, to hair and fur. And what you do is you go through and check off who has what, and you can create a tally of most common to least common traits. And then what you're going to do is take that chart and convert it into a stacked Venn diagram. So the largest circle, and I know it's not quite a circle on my screen, you're going to put the trait that is the most common. It's the one shared by all, so that is why it's the biggest circle. Then inside of that circle would be the next most common trait. It's shared by 5 out of the 6. So that means there's one organism that has a vertebrae, but it doesn't have 4 limbs, so it's not going to go into that circle. Like, we could put the names of the organisms in the circles to really help us visualize this. And then inside of the 4-limb circle would be the next most common trait, which in this case was the amniotic egg. It was shared by 4 out of the 6. But then for the next 2 traits, they were equal. It wasn't like it went 3, then 2. So because of that, I have to put 2 separate circles inside of the amniotic egg. There's one for the eggshell trait and one for the hair fur trait. And the reason these are separate circles is if we actually think about the organisms that we're dealing with here, the ones that have hair and fur don't have eggs with eggshells. So they need 2 separate circles. And then once we have this stacked Venn diagram, you can already start to see the cladogram taking shape. So you're going to convert that Venn diagram into the branching cladogram. So the main line here is where you're going to put all the different traits that you're comparing, and then you'll put your branch points where they belong to indicate the different species that you are comparing.